So very good morning to one and all present here. With the blessings of our Honorable Founder President Sir, Dr. Ashok Kumar Chauhan Sir, and the inspiration from our Chancellor Sir, Dr. Atul Kumar Chauhan Sir, today we are going to organize one-day workshop on cytogenetic analysis in cancer diagnosis. So cancer is not only a worldwide problem, along with its health issue, it also has economic, social burden on us. So here with this workshop, we will try to pursue the all the techniques that how the cytogenetics can lead to early diagnosis of the cancer, and this will be very helpful to prevent the cancer by early treatment processes. So for this occasion, we have Dr. Tahir Moidin Mala sir, who is postdoctoral research associate from the Department of Endocrinology, Seri Kasmin Institute of Medical Sciences, Srinagar. So before moving forward, we are well blessed with our Honorable VC sir with us. He is expertise in the field of physics, and the physics has lots of things to do with cancer. So, sir, please this occasion with your words of wisdom, sir.
Thank you very much, Dr. Kunal, Dr. Datta, and the lady who was comparing. I forgot
Dr.
the name.
Rajni. Dr.
Rajni. Yeah, sorry for that. And that's the type of cancer that will come when you grow a little old. You start forgetting names as well. And speaking on this is not my forte at all. So I would rather welcome the speaker, the keynote speaker, Dr. Tahir, for sparing his time and expertise to give this exposure for one full day to our students, our faculty members. Of course, this is an area which is not only dreaded or maybe people fear from this, but there are ignorances also prevailing all around. There are misconceptions, myths around what cells may be cancerous, what may not be cancerous, what grows as a cancer, what can be curated at early age, and then to what stage it is. It would be a nice exposure for all of us to really understand aspects. And I'll not be able to comment on any of these specific topics in particular, to be very frank, even though you said that physics is related to cancer. Well, it happens on physical part of the body. That's it. That's biophysics may be some part related, and maybe some understanding may be there. I'm not at all from there. So my best wishes for the day. If you want me to speak, I would speak on particle physics, and rather than cosmology, astrophysics, that may be my area of expertise. My best wishes once again. Over to you, and enjoy the day with cancer. Thank you. Or
Thank
without
you so
cancer.
much, sir.
Thank you.
Thank you, sir. Thank you so much, sir. So because of some unavoidable condition, our respected director, sir, is not here in this workshop, but he has conveyed his blessings and welcomed you, sir, Dr. Tahir, sir. He has welcomed you. Now, I may request our respected registrar, sir, Mr. Prabhakar Tripathi, sir, to place this workshop with his inspirational words.
Thank you, Dr. Rajni, madam. I once again welcome Honorable VC sir and respected keynote speaker, Dr. Tahir Moeddin Malla sir, and all our faculty members and all dear students. Of course, this is a very good topic which our department is making awareness program, like so that all the audience should know that before coming to this in a very dreadful situation, one should how can get rid of it. Because I came to know that so many Bollywood stars, despite following their dietitian instructions and very meticulous in their eating pattern and all these practices, but they unfortunately became the victim of cancer. So understanding that what causes the cancer and how this can be avoided. So these programs are certainly very much beneficial, not only to students also, but to the mass community also. So my heartfelt wishes to the organizing team of biotech department that through the expert talk of Dr. Tahir Muhaddin Malla sir, many of our students would be benefited, not only general understanding, but also they are biotechnologically fit also. So my best wishes to the organizer, my sincere thanks.
Thank you so much, sir. So our PC, Dr. Vishnu Samanta sir is also here. So sir, if you can share a few words on this workshop.
Yeah, ma'am. Yeah, thank you. Thank you. Actually, we are already getting late. So just I would like to extend a warm welcome on behalf of MIT University Jharkhand Ranchi to our today's guest speaker, Dr. Tahir sir. So warm welcome to this MIT platform. And I would also like to thank the department for organizing that webinar and on this topic, I mean, cancer diagnosis, early cancer diagnosis. So I hope that student will get maximum benefit out of this session. So with best wishes and again, warm welcome to the speaker, today's speaker, and I hope the student will enjoy the session. Thank you. Over to Rajini, ma'am.
Thank you, sir. So I, Dr. Rajni Sharma, on behalf of Amity Institute of Biotechnology, Amity University, Jharkhand, once again welcome you, sir, Dr. Tahir Muhaddin Malla, sir. So before hearing you, sir, I would like to introduce you to all the audience. So Dr. Tahir Muhaddin Malla, sir, is PhD in genetics and presently working as postdoctoral research associate at Department of Endocrinology, Seri Kashmir Institute of Medical Sciences, Srinagar. He is a life member of Indian Society of Human Genetics. He is the recipient of Young Scientist Award in Medical Sciences and received many Best Posters awards in many conferences. He has more than 20 publications and attended more than 20 national and international conferences. So, sir, it will be very honor to us to hear you over the particular this topic over cytogenetics. So, over to you, sir.
Thank you so much. I, at the outset, would like to thank the organizers, the Vice Chancellor of the university, the registrar, sir, Dr. Kunal, Dr. Rajini, Dr. Amit Datta, sir, and Dr. Biswaroop in particular. I thank you all for providing me this opportunity to share my experiences about cytogenetic analysis in cancer diagnosis. I'm sure I'm very much audible to you. Is it?
Yes, you are audible, sir. You're audible. You're audible and your screen is also visible. Please carry on.
So my presentation will be all about cytogenetic analysis in cancer diagnosis. As you all must have heard about cancer, I will not talk much about what cancer is and how it progresses. You all know that it is an uncontrolled division of any cell, maybe a blood cell or a tissue organ related cancer. So I will go to the basics so that the students, majority of the students, understand what cytogenetics is all about. The cytogenetics basically deals with the genetic constitution of cells through the visualization and the analysis of the structure and the properties of chromosomes. Chromosomes, you must have heard that every cell has a nucleus, and in the nucleus there are chromosomes which we inherit from our parents, which everyone inherits from his or her parents. So we have a diploid number of chromosomes. Every human has a 46. Every normal human being has a 46 number of diploid chromosomes. So these are present inside the nucleus, and actually these structures, they only harbor almost all the DNA that we have. So every information that is inherited from parents is through these chromosomes only. When the mitosis and meiosis takes place, and after the meiosis we get sperms and ova, and after the fertilization we get these chromosomes inherited. So knowing about the genetic constitution of cells through the visualization or the structure and
Tahir, sir.
properties of chromosomes.
Tahir,
Yeah,
sir,
yeah.
I little bit interrupt you. Sir, kindly please make it slideshow mode, and your slide is not moving. You are already in first slide.
Okay. So I was talking about what cytogenetic basically means. Cytogenetics is the analysis of chromosomes, their structure, the properties of chromosomes, their behavior during mitosis and meiosis, and their influence on the phenotype or of an individual or a disease, or what influence they have on a particular disease, and the factors that cause chromosomal changes. So all these, all this is studied under a subject that's called as cytogenetics. So there have been some landmarks in the history of cytogenetics. I'll go through these. The chromosomes for the first time were coined, the term chromosomes was coined by Walder in 1890. Then the experiments that were carried out by Sutton, they said that the every inherited character, it comes through the chromosomes. That's called as chromosome theory of inheritance. And after that, Morgan made some developments, and he said that the genes that are responsible for a particular character are located on chromosomes. And after that, the exact number of human chromosomes as 46, it was revealed by Tijo and Levon in 1956. It is then only subsequently many of the syndromes were detected and discovered, like in 1959, trisomy 21, which is the most common chromosomal anomaly in humans, which is called as a Down syndrome, having an extra copy of 21st chromosome. And then subsequently, Turner syndrome, having a single X, females having only one X chromosome, called as Turner syndrome, it was discovered. Then Klinefelter syndrome was revealed as double XY males, which have an extra copy of X chromosome. So since then only, it's well established that these chromosomal abnormalities, they contribute to the genetic diseases and other related phenotypes. And after 1980, with the advancements in molecular biology and genomics, and with the integration of techniques, significant advances were made in understanding the genomes throughout the chromosomes. So nowadays, with the advances in microscopy, the application of bioinformatic tools, and the integration of chromosome analysis and genomic data, it has proved to be very fruitful the future of cytogenetics. So these are some of the landmarks, and these are all the techniques about cytogenetics, how we do cytogenetic in chromosomal disorders, or in particularly cancers. So number first is the conventional cytogenetic analysis. Conventional cytogenetic analysis is just visualizing these chromosomes after a proper staining and banding procedures under a microscope. And it's called as karyotyping as well. Then we have interphase FISH. Interphase is a stage of the cell division. These nuclei are subjected to fluorescent in situ hybridization. Then we have whole chromosome painting FISH. Then we have a multiplex fluorescent in situ hybridization. We have multicolor bands. We have advanced technologies like chromosomal microarray, or this is also called as array CGH is one another technique. These are almost with a slight difference. This genomic hybridization and array genomic hybridization. These are two more techniques, advanced techniques, which are followed in the cytogenetics and in the diagnosis of cancers. So the number first is the conventional cytogenetic analysis, which is the most widely used technique for diagnosing the cancers. Sometimes we deal with leukemias and other solid tumors. But what this conventional cytogenetic analysis is, as you all know, we have cell division, which has many phases like prophase, metaphase, anaphase, and telophase. And when the cell is at rest, it's called as an interphase. So after the cell is stimulated, or a bone marrow cell takes a division, cell division, the cell passes through prophase, where this chromonemata, or the genetic material that is present in the cell, is condensed and coiled, and it forms a rod-like structure. And later on in the metaphase, this gets very much visualized, and this is the chromosome. It gets into its shape, actual shape, where it can be identified and it can be isolated from this DNA, from this nucleus, sorry. So these chromosomes are visualized under a microscope. So how do we do this process? I'll let you know in the next few slides. So what are the samples that can be used for this conventional cytogenetic analysis, or even other cytogenetic techniques? The most preferred samples are peripheral blood, which are used for genetic disorders and some leukemias. The number second sample is bone marrow. Bone marrow is again a preferred sample for leukemias, and in some perinatal diagnostic procedures like amniocentesis, we use amniotic fluid. It can also be used for conventional cytogenetic analysis. And if it is a cancer of some organ like breast, or some other lung or other organ, we can use tissue samples as well. So this is a diagrammatic representation of how we proceed with a sample. Once we get a sample, bone marrow or blood sample, we incubate it in a medium and at optimum temperature and carbon dioxide concentrations. We proceed further. We incubate it in a carbon dioxide incubator for almost 24 to 48 hours, if it is a leukemia sample, cancer sample. And then we arrest these cells on a particular stage that's called as a metaphase. So finally, we get these cell suspension fixed by using a fixative, and we proceed further by making a slide with an airdrop method. We drop some drops of the cell suspension onto a slide, and further staining is followed, and microscopy is done. And finally, the slide is screened under a microscope using a software or a karyotyping workstation, and these chromosomes can be arranged into a karyotype. That gives you the final diagnosis. So how do we do that? This is the technique. We take 1 to 2 ml of heparinized blood. It should not be clotted. Bone marrow or blood can be used. And we use a specialized kind of media that's called as RPMI 1640. It's the most widely used cell culture medium for lymphocyte as well as bone marrow cultures. Although now we have specialized media for bone marrow known as bone marrow max samples, which have got specialized factors, growth factors, so that these cells, cancerous cells, they grow in optimum conditions. And if we are using a blood sample, as blood cells are non-dividing cells, we need to initiate cell divisions into them. So we use a kind of it's a mucoprotein, a plant product, phytohemagglutinin, that is extracted from Fasciolus vulgaris pulses. So it can be extracted from them. It is commercially available also. So this initiates the cell division in vitro in the medium once we incubate these cells in the CO2 incubator. And at 37 degrees Celsius for 24, 48, and 72 hours, these cultures can be carried out. But we always set up parallel cultures that are initiated for every patient so that we don't miss any chance to get the clone, the abnormal chromosomes out of these cells once we report. And in cancers, we can have different types of cultures that can be direct, overnight, short-term, and long-term cultures. So these are types of cultures that can be done for cancers. But for other genetic disorders, common genetic disorders, we usually do long-term 72-hour cultures and arrest the cells by colchicine at 70 hours. So on day 2nd and day 3rd, if it's a long-term culture, we check these samples for contaminations, as all this should be done in aseptic conditions. Once if the cells are contaminated, cultures are contaminated, then it is very difficult to report these and to get chromosomes out of these cells. So on day 4th, the harvesting of these cells is done at 70 hours. We add a solution, colcimid. It is again a plant product, colcimid solution that actually inactivates the centrioles, and the spindle fibers are not produced in the metaphase cells, and these metaphase chromosomes, they lie inside the nucleus. And once we get these cells after centrifugation, we incubate them with a hypotonic solution of potassium chloride, and then this is used for to imbibe these cells so that these cells get swell up. And after that, we fix these cells using a cornized fixative, which is again a combination of acetic acid and methanol, and we repeat washing cells with this fixative, and finally we get a cell suspension, and this cell suspension is dropped onto the slides. And once we drop these cells from a height on the slide surface, some of the nuclei which are in the metaphase and are imbibed well, they and are swelled up, they get bursted and the chromosomes come out. So once these chromosomes come out, these slides are stained using GIMSA stain, and the banding is performed. Banding is done by using a trypsin solution and GIMSA stain. So it's called as GTG banding. And after that, finally, we do the microscopy. Microscopy is done under 10x objective of the microscope so as to locate these metaphases, these small bunch of chromosomes. You can see some small groups of chromosomes lying between these cells. These are the metaphases, and then we focus on a well-spread metaphase, finally getting a larger picture under an 100x objective of the microscope. So this is how we process these samples. So once we process, we karyotype this metaphase, and we locate these chromosomes and compare them band for band, making use of imaging software which is attached to this microscope. So how we do this, I'll let you know. This is a human karyotype where you can see these are 46 chromosomes, a female karyotype. And how we do a karyotype is using a software, we pair up these chromosomes, as you can see, from 1 to 22 pairs, and then X and X. These are the sex chromosomes. So these chromosomes are paired up and arranged on the basis of 3 criteria. Number first is the length of the chromosome. So the largest ones are placed on top in group A. So these are large metacentric chromosomes. Metacentric means where the centromere is present in between the chromosome, almost halfway. So these are large metacentric. Then we have B group chromosome 4 and 5. These are large submetacentric. Then we have C group chromosomes, which are medium submetacentric chromosomes. Then we have D group chromosomes, where centromere is present at towards the end of these chromosomes. They are known as acrocentric chromosomes. So based on the position of the centromere and the length of these chromosomes, we group these chromosomes into different groups. So 16, 17, and 18, these are small submetacentric. Then we have small metacentric chromosome 19 and 20. This is F group. Then we have G group, again an acrocentric chromosome, where these are small acrocentric chromosome, 21st and 22nd chromosome. Then we have X chromosomes. It can be, in case of a female, it can be XX, and in case of a male, it can be X and Y chromosome. So this is how we make a karyotype. So let me tell you here what these bands mean actually. These dark and light bands are produced after we treat these chromosomes with trypsin solution, which is a proteolytic enzyme. This trypsin digests these and digests these chromosomes differently in different areas. So these, once we stain this with the GIMSA, the regions that are condensed, they retain the stain. So they are the dark regions, dark bands, and the regions, those do not retain the stain, they are lighter in color. So these are called as light bands. So it is known that these darker regions are adenine and thymine rich regions, AT rich regions, and these light regions, they are GC rich regions. So these regions are all actually they are possessing the exons. Most of the transcriptional activity is actually happening in these light areas. So the light areas are more important than the darker regions. So this is how we compare this band for band. As you can see, every pair has unique banding patterns. So once we get a karyotype, we find that this cytogenetic abnormalities or cytogenetic diagnosis, it has many applications. So how does it help in cancer? It helps in to establish the presence of a malignant clone. Malignant clone means an abnormality in chromosomes. It can be a translocation or a deletion. And to clarify the diagnosis, as many cancers have mixed phenotypes, where you can have in the complete blood picture, you can have different, you can have similar blood pictures, but to confirm that as to what type of the cancer is, cancer cytogenetics plays a very important role. And after we get the diagnosis, cytogenetic workup, we actually help the clinicians to indicate a prognosis. It can help in deciding to what treatment should be given to this patient having this kind of a chromosomal abnormality. As in cancers, there are varied types of chromosomal or genetic changes, wherein a clinician needs to look into that what type of the chromosomal abnormality or a genetic mutation is present, and what it corresponds to, as there are some favorable genetic mutations and some unfavorable genetic mutations that can be detected. Likewise, in cytogenetics also, there are many cytogenetic changes or chromosomal abnormalities that help a patient in a way that they respond differently. They respond in a good manner to a chemotherapy or to a drug, rather than having another chromosomal abnormality that will be having a poor prognosis. That means the outcome of the disease will not be good. It will not respond to the same chemotherapy in a similar fashion as the other chromosomal abnormality gives it. So different chromosomal abnormalities, patients having different chromosomal abnormalities, they respond to the same chemotherapy in a different manner. So this helps clinicians to decide as to what treatment strategy should be decided for a particular patient having a particular chromosomal abnormality. So it also helps in to monitor the response to the treatment. So once we detect a chromosomal abnormality, for example, in case of chronic myeloid leukemia, if it is having a 922 translocation, so once a clinician treats the patient with some chemotherapy or with a particular drug, after completion of the therapy, again we do a cytogenetic analysis of the bone marrow, and we see the remission of the disease as it can be detected that how many percent of this chromosomal abnormality is reduced, or is there any abnormality present or not. If it is not present, it means it is in the remission phase. It has responded well to the chemotherapy. So it also supports for further research in this area. So how do we identify clones? Let me tell you, this is one of the patients having chronic myeloid leukemia. It is a cancer of a particular blood cell, a myeloid blood cell, a common blood disorder. So what is the diagnostic abnormality is, or a clone that we detect, is 922 translocation. So simultaneously, when two chromosomes break up, they break down due to some reason which can be known or unknown. So the fragments or the material of two chromosomes is exchanged. So once these genetic material or the chromosome fragments are exchanged between two chromosomes, it produces a translocation. So in this case, in case of chronic myeloid leukemia, we have a 922 translocation where some of the material from genetic material or chromosome fragment is a band is translocated to the 22nd chromosome, which is known as also known as Philadelphia chromosome. So this is how we detect it on a karyotype. You can see if you compare chromosome 9 pair, so you can see the right side chromosome is a bit larger, and the lower side band, which is indicated with an arrow, Q3 to 4 band, it is in addition, it is extra in this right side chromosome 9. So this is an abnormal chromosome 9. In chromosome 22, at the downside, you can see indicated by an arrow, it is a shorter chromosome, very small 22nd chromosome. It means it has lost some area, it has lost some region. So this region, what is lost on chromosome 22nd, has translocated to chromosome 9, so producing a shorter 22nd chromosome, which is known as 22nd chromosome. So where this break happens, so at the Q3-4 region, we have BCR, and at the Q11 region, we have ABL. So these are two genes which once they get fused in the Philadelphia chromosome, producing an ABL BCR fusion gene, which produces ABL BCR protein that actually results into development of this cancer, type of blood cancer. So again, this is another type of blood cancer, acute promyelocytic leukemia. It's the leukemia of promyelocytes, and this is diagnosed by looking into the karyotype. If the patient is having a translocation of 1517, translocation between chromosome 15 and chromosome 17. So this makes the diagnosis of this blood cancer. So I'm trying to let you know how we diagnose these. and what exactly these translocations are. As you can see on the left side of at the position 15, chromosome 15, the left side chromosome is a normal one having a single single dark band in the middle area, and while this right side 15th chromosome has two dark bands, the lower side it has an additional dark band. So where from this has come? This has come from this chromosome 17. On the right side you can see this 17 is smaller than the left side 17 chromosome, which is the normal one. So right side chromosome is the abnormal one, where some this lower band has translocated to 15. So giving rise to a again the fusion of two genes, PML, promyelocytic leukemia gene, and rare alpha, that is retinoic acid receptor alpha gene. So once these two genes they get fused to each other, they produce a malformed protein, a protein that is not very good, and that protein actually, that fusion protein that produces and leads to the development of this blood disorder, blood cancer. So this is how we identify these clones. I will let you know how we identify them with different techniques, other techniques also in the next coming slides. But let me tell you how it helps in the disease prognosis, how we conclude anything from these chromosomal abnormalities. Like in acute lymphocytic leukemia, if we do some cytogenetic analysis and we find these hyperdiploidy. Hyperdiploidy means if we have more than the diploid number of chromosomes. If we have more than 46 chromosomes in the patient, it is a favorable. It has a favorable prognosis. Means this patient will respond to the chemotherapy efficiently. And again, this translocation 1221. If we detect this translocation by cytogenetic analysis, it is again favorable translocation. This has a good response to the chemotherapy. So trisomy 4, trisomy 10, trisomy 17, again, these trisomies also have favorable response to the treatment and have favorable outcomes. So what are the non-favorable ones? If it is Philadelphia positive, like what I was telling you in an earlier slide, that if it is a 922 translocation in acute lymphocytic leukemia, it has a bad prognosis. It means it does not respond well to the treatment and the disease outcome is not that good. So again, if we move further, hypodiploidy means if it is less than 46 chromosomes, like it is 44, 45, if it is not a normal diploid number of chromosomes in a patient in some cells, so it is not a favorable condition for him. Again, if it is a translocation of 411, again, the prognosis is not that good. So this is how cytogenetics helps clinicians to decide and to know about the prognosis and the outcome of the disease in the earlier stage. Once the patient reports to a clinician and a clinician suggests a cytogenetic analysis, he comes to us. So once we get these karyotypes and cytogenetic analysis done, it helps patient as well as the clinician in deciding what to do and what chemotherapy to follow. So favorable anomalies, chromosomal abnormalities will definitely be beneficial to the patient. So we have many other diseases like myelodysplastic syndrome, MDS, or it's a pre-leukemic stage. It can further develop. The patients with myelodysplastic syndrome, they can develop AML further in future if they are not treated at the right time. So this MDS and AML, they also can be subjected to cytogenetic analysis. And if we find these translocations like 1616 translocation or inversion 16 or translocation 821, these are favorable, or translocation 15-17, which I was discussing in earlier slide in the acute promyelocytic leukemia. So this is also a favorable chromosomal abnormality in these cases. Then we have unfavorable ones like deletion 5, deletion 5q, or deletion 7, or deletion 11q23, means deletion of the 11 chromosome. Then translocation 619 or translocation 922. So this is how these translocations or chromosomal deletions, they help in establishing the prognosis of these patients. But although this is a widely used and the basic genetic test that is done for these cancers, this has some limitations. Let me tell you what limitations are there in the conventional cytogenetics. This can only detect chromosomal defects that are larger than 3 MB size, 3 MB of DNA. If it is less than 3 MB DNA change, it cannot be detected by conventional cytogenetics. Then the next challenge in conventional cytogenetics of cancers is that the culturing cancer cells and getting metaphases is a very tedious and a cumbersome process. Not always you get good morphology chromosomes, and these cultures often result in poor morphology chromosomes that are difficult to analyze. So if the chromosome morphology, chromosome structure is not that good, so the chromosomal abnormalities and the karyotypes cannot be made efficiently. So it will be a challenge to the cytogeneticist. And again, it is time consuming. If we are going for long-term cultures and a clinician needs a cytogenetic analysis, or he needs to put a patient to some chemotherapy, so he needs to decide on that. So conventional cytogenetics takes its time, like it can take 72 hours or more than that also. So how to overcome these challenges? There have been many other techniques, like fluorescent in situ hybridization. It's a technique that can identify cryptic translocations. Cryptic translocation means hidden translocations which cannot be otherwise detected by conventional microscopy or conventional cytogenetics. So this is another advanced cytogenetic technique that is in use, and that can be applied in a to diagnose these patients in a better manner. So how do we do this fluorescence in situ hybridization? I will just read out the steps. And we use the SAM sample, like lymphocyte cultures or bone marrow samples can be used. We can directly process these cells by hypotronic treatment and coronarized fixative, as I discussed earlier. It can be cultured also, and these cells can be processed directly also. So we then use washing procedures. We wash these cells at 73 degrees Celsius by 2x SSC, and then we treat it with protease at 37 degrees Celsius. Again, we wash it with 2x SSC. Then we dehydrate these slides with dehydrating solutions, ethanol at different percentages. And then finally, we get these slides and we add a probe mix. Probe is a specialized and a particular DNA that is labeled with a fluorescent molecule. So that is used in this technique, and ultimately that probe is annealed and hybridized simultaneously with the slides with using a hybridizer. You can see this is a hybridizer wherein we put our slides, and once it is hybridized, these probes are hybridized, we move further and analyze these slides under a fluorescent microscope. So what exactly is the probe that is used to target a particular stretch of the chromosome or a particular stretch of the DNA? So in fluorescence in situ hybridization, what we do is we use DNA probes. DNA, you all must be knowing what DNA means. And we target a particular locus on the chromosome, or a particular sequence of DNA inside a chromosome. So if we have a probe DNA that is labeled with the fluorescent molecules, it can be any color, it can be green or orange. Here it is orange. So this fluorescently labeled molecule or probe, this is denatured simultaneously with these chromosomes on slides or interphase nuclei, which are present. The chromosomes ultimately are present in the nuclei. So we need not to get these chromosomes out of the nucleus, but they can be the cells can be directly processed and can be denatured with the labeled fluorescent probes. So once the probes are hybridized with these nuclei, you can find if that particular probe DNA has a complementary sequence on the particular chromosome. It means it is present, that particular stretch is present on the chromosome. If you find no signal, no orange signal in the nucleus under a microscope, it means the region of the chromosome is absent, means it is deleted. So this is how we detect a deletion or a translocation using a fluorescently labeled probe. Presence means it is present, and the absence means deletion. These probes can be DNA or RNA that is complementary to certain sequence on the target DNA. These there can be plasmid phage DNA or cosmic DNA or plasmid DNA probes, which are commercially available. You need not to manufacture or make them in the laboratory. So for every translocation, for every deletion that is related to some diagnostics, there is a commercially available probe, which you just need to buy that probe and then use it on your slides. Or it can be a probe, can be a PCR product also. So this is a diagrammatic representation of a probe. If it is a specimen DNA of the patient or it is a chromosome, so it has some nucleotide sequences, and complementary sequences can be made, which are labeled with fluorescent molecules. So this will be used as a probe. So this is how a probe is. So there are different types of probes which can be used to actually diagnose different types of chromosomal abnormalities. We have first the centromeric probes, we have telomeric probes, we have whole chromosome painting probes, and locus specific chromosome probes available. The centromeric probes, they just identify the centromere of the chromosome. It means if the centromere is detected, means the chromosome is present. If the centromere is not detected, means the chromosome is lost. It means it's a monosomy. For example, if we use a probe for the centromere of chromosome number 1, and if that signal is not present in the nucleus under a microscope, it means the chromosome first is not present there, it is lost. So likewise, if we need to detect a telomere, so we can use telomeric probes, or we can paint this chromosome. Whole chromosome can be painted using a whole chromosome painting probe. So this is how using these different probes give us different pictures under a microscope. If we use a locus specific or a centromeric probe, this is how the picture will look like. So these are all chromosomes, and this orange signals are centromeres. It means we have used centromeric probes for these chromosomes, for a particular chromosome. So I'll move further. I'll not take much of your time. This is a locus specific probe. This is a whole chromosome painting probe. So this is how it will look like under a microscope, the images. So this is how fluorescence microscopy is done. This is a fluorescent microscope attached with a software. We use different filters to locate different colors of probes. It's attached to a CCD camera. Then it is attached with a fish analysis software. So this is like if it is this is a CML patient or a CLL patient, and we need to know about 17th chromosome P53 gene. So for detection of genes, presence of genes or the deletions of genes, we can use fluorescent in situ hybridization. Like you can see, there are only 3 signals, while it should be 2 green and 2 orange signals. Loss of 1 orange means loss of P53 gene. This is how we detect using fluorescent in situ hybridization. This is in case of breast cancer. You can see HER2 DNA is targeted, and there is an amplification of HER2 DNA in breast cancer patients, and this is the progression. While the progression of the disease happens, this HER2 DNA amplification takes place. So we detect it using orange color and green color probes. You can see there are 2 green, 2 orange cells. It's a normal cell, and this is a HER2 positive amplification, where you can see a lot of orange signals. It means lot of HER2 DNA is present there. So this is the amplification. This is how we detect different rearrangements. We can use different kinds of probes to locate different kinds of chromosomal abnormalities. This is our workstation where we detect these chromosomal abnormalities and different translocations for different cancers. Right side is the list. These are kinds of probes that we use, and you can see I have shown this karyotype to you, 922 translocation, how it can be detected by using fluorescent in situ hybridization. You can see a fusion signal, orange signal, 1 orange, 1 green, and 1 fusion. Fusion means BCR-ABL fusion has happened. 922 translocation has taken place. So this will be a positive cell, a positive patient for BCR-ABL fusion. Again, we have ETV6 RANX1 in ALL patients. So this will be a fusion. There will be a fusion signal in addition to these 1 orange, 1 green signal. There is an orange mixture of orange, orange and red and green signal. It will give an orange signal. So orange signal means a fusion. Again, we have many other deletions that can be detected using FISH also, like loss of orange signal in these cells, in this cell. So this is deletion 5. So this is a normal 2 orange, 2 green. You can see this is by karyotype. You can detect this deletion, and this is by fluorescence in situ hybridization. Again, many using whole chromosome painting probes also, which give different colors to different chromosomes. You can see a 922 translocation. 9 green color can be seen present here on 22nd, and 22nd red color can be seen at 9. So this is a translocation detected by multiplex FISH. Then we have 2 advanced techniques. I will wind up. Advanced techniques are array comparative genomic hybridization. Comparative genomic hybridization means where we actually compare the test DNA and a normal DNA. So this is a latest advance in the chromosomal studies, where you can find out and detect the losses and gains of chromosomes. So how we do this is we take the test DNA and the control DNA, we label it with different fluorescent dyes, and there are 2 methods. Comparative genome hybridization, we use a slide, a normal chromosome complement, and then we put this test DNA and the control DNA, we hybridize this to this chromosome plate, and then we find the comparative colored regions. If it is only green, means the test DNA is increased. It means it is gained. It's a gain of the DNA material there. at the region, green region, and at the red spots, it will define that it is a loss of the DNA material. Likewise, we have arrays. We have a different oligo arrays which are in the market commercially available for different cancers, different diseases. We have different arrays, and hundreds of regions can be analyzed simultaneously. Whole genomes can be analyzed simultaneously using these arrays, wherein we use the test DNA and the control DNA onto a commercialized chip, and then we analyze using a bioinformatics tool and a software which gives you different spots. It means the orange spots which are brighter in color means gains, and which are lighter in color, those regions are losses. So losses of DNA and gains of DNA throughout these chromosomes or throughout the genomes of a person can be detected using these techniques. So to conclude, in this era of array diagnostics, cytogenetic techniques have a great role to play in the diagnosis of cancers. Cytogenetic techniques, they not only help in detecting the etiology or the cause of the disease, but also in understanding its outcomes. And if we follow the integrative approach, it helps in detecting cryptic translocations which are otherwise undetected. And advanced molecular cytogenetic techniques like chromosomal microarray, it helps in detecting losses and gains of DNA throughout the genome at one go. So I thank you once again for providing this opportunity to me, and I will be happy to answer some questions if anybody has some doubt. Thank you once again.
Thank you so much, sir. So there are two questions. One question got self-answered while you are explaining the slides. One question is from Dr. Dhananjay Kumar Pandey, sir. He first thanked you for your informative talk and asking, I have just a quick query that what are possible reasons behind favorable prognosis and chemotherapy with hyperdiploidy trisomy and specific translocation in chromosome? Should I repeat, sir?
Yeah, I got the question. So there are translocations or particular chromosomal abnormalities. As I was telling you, I could not explain it, I think, in a broader manner due to lack of the time. So there are translocations that lead to fusion of two genes, two chromosomes. Once the segments of two chromosomes, they translocate, they exchange their positions. The two genes that are fused while this translocation takes place, it produces a different kind of a protein, fusion protein. So in some diseases where some genetic mutation or this DNA mutation or of a gene or a fusion protein is formed, this kind of in the subsequent molecular pathways or biochemical pathways, this helps once a particular drug is given to the patient as a chemotherapy. So this acts as a kind of it provides an environment, a favorable environment. That protein that is formed, it provides a favorable environment in some or the other pathway it takes part, and it plays its role in the better response, showing a better response to that particular treatment.
Thank you, sir. So those who will be having any query, we will transfer to you, sir. So very quickly, I'm now inviting Dr. Amit Kumar Datta, sir, to give a vote of thanks.
Very good afternoon to all. At the onset of blessings of Honorable Founder President, Dr. Ashok K. Chauhan, sir, and the inspiration from our Honorable Chancellor, Dr. Atul Kumar Chauhan, sir, we are very much thankful to our ever supporting Honorable Vice Chancellor, Professor Dr. Raman Kumar Jha, sir, respected Director, Professor Dr. Ajit Kumar Pandey, sir, respected Registrar, Mr. Prabhakar Tripathi, sir, for allowing us to conduct this workshop on cytogenetic analysis in cancer diagnosis. We, on behalf of Amity Institute of Biotechnology, would like to thank today's expert guest from Seri Kashmir Institute of Medical Science, Srinagar, Dr. Tahir Mahiuddin Malla, sir, for his very precise and informative session on how to diagnose different cancers through advanced cytogenetic analysis. Once again, thank you, Dr. Tahir, sir, for accepting our invitation and sparing time from your busy schedule. We are also gratefully acknowledged to our respected program coordinator, Dr. Bisharup Samanta sir, for his kind help and support to organize this workshop. We also like to thank all the university coordinators and program coordinators, sir and madam, especially Dr. Jaika Chattopadhyay ma'am, for their cooperation and support. We also thankful to all the student participants of Amity University, Jharkhand, and from the outside institution of Amity University, those who joined with us in this workshop on cytogenetic analysis in cancer diagnosis. We owe our deep gratitude to all the faculty and staff members of Amity University, Jharkhand, and others who joined in the workshop. We are also thankful to admin and media team who have supported us for conducting such valuable sessions. We are very much thankful to the IT hosts team Noida, especially Mr. Arvind Mitra ji, for providing smooth IT platform for conducting this workshop. Without the support of IT team, it was not possible to conduct this such a valuable session. Last but not the least, on behalf of Amity Institute of Biotechnology, we also thank to Dr. Rajni Sharma ma'am, moderator, and Dr. Kunal Kumar sir, co-convener of this workshop, for their all-time help, support, and cooperation. Thank you, sir and madam, and thank you very much to all once again to join with us. Over to Rajni ma'am.
Thank you so much, sir. So once again, I am thanking Dr. Tahir sir for delivering such a nice speech to us and how the cytogenetics can be used for the early diagnosis of the cancer. It was really very nice session, sir. So, sir, if you permit, then we can end up the session.
Yeah, thank you. Thank you so much.
Okay. So if Dr. Samanta sir permit us, then we can close the session.
Yes, sir. Yes, sir. Please conclude the session. Thank you.